How are you doing? It's so nice to see you.
Great.
I'm wearing your T-shirt from 2 years ago.
Great.
I had such a good time. It was so wonderful.
Yeah.
So
Excellent.
I hope in another 2 years we can meet in person again.
I will hope the same.
Yeah, that would be wonderful.
How is Belinda?
Everything's okay. Everything's great. Yeah.
Great.
So yeah. So I excited to present my research today because it's the first time that I'm giving this talk. Is
great.
Fresh, so to say.
Fresh.
Not even as a preprint. So everything's completely new.
Nice.
Yeah.
Sebastian,
It's a
good
long.
afternoon. Dr. Vinod
Hi.
here.
Hello.
Hi, Sebastian.
Good. Just let me know when I should share my screen.
Sure.
Yeah.
Hey, Sebastian, just give me 2 minutes time now.
Yeah. Okay.
Actually, Prashant is here. He's in the lab.
Hi, Prashant. It's so nice to see you. Yeah.
Hi, Sebastian.
So no one's wearing the T-shirt from 2 years ago. Yeah. But you look very good.
Yeah.
Good. So.
Ramesh?
Yes, sir.
Ramesh?
Yes, sir.
Yeah, I have shared the link to you.
Okay.
Which link? Live?
Yes, live link I shared.
Okay. Yeah.
Hi, Sebastian. Good to see you.
Live.
Nice to see you. It's wonderful.
இல்ல நம்ம யூடியூபுக்கு காலையில் நினைச்ச மாதிரி அதே
Archana, are you there? Archana?
Sir, Archana is not there, so I think Sandeep may take say yes.
Okay. Sandeep, you can start. Sandeep, ma'am.
Yes, sir. Welcome all to the post-lunch session. Moving further, it is a great honor to have with us our next speaker, Dr. Sebastian Lepton. Dr. Sebastian is presently serving as Associate Professor at Zhejiang University, University of Edinburgh Institute. He completed his PhD from University of Munich, Germany, and later served as postdoc fellow at University of Oxford, National University of Singapore as well, and served as professor without chair at University of Hohenheim, Germany. The Lepton Lab focuses on bacteriophage biology, phage therapy, synthetic phage biology, membrane proteins, etc. He has till date more than 60 research publications and many completed and ongoing projects with collaborations with many reputed institutes worldwide. Thank you so much, sir. May I now request Dr. Sebastian to share his expertise with us on the topic elucidating the molecular basis of phage resistance in colistin-resistant Acinetobacter baumannii. Over to you, sir.
Thank you so much for this kind introduction. Let me just share the screen. Yes, that's the one. Okay, good. So first of all, I would like to thank you a lot for the kind invitation and that I can present this research on elucidating the phage resistance mechanism of a strain that is colistin resistant of Acinetobacter baumannii. We previously heard that there are not many colistin resistant Acinetobacter strains in India, but with my collaborators at the Shaw Hospital, Acinetobacter baumannii and Klebsiella are the most important pathogens in the clinic that cause a lot of trouble in old people and immunocompromised people, and of course in patients that had major surgery. So 2 years ago, why is my screen not? 1 second. Something's wrong. I can't.
Sebastian, can you minimize the screen? So
Yeah,
we'll
one
check whether
second.
it's-
Yeah. Now it's at the moment it's not working at all.
Sometimes if it is in full screen, it won't work. You can minimize it. Not full screen mode.
Okay, okay. Good. Then I have a few animations which won't work, but that's okay. Okay, can you
Yeah, you can take your time.
can you see my screen? At the moment you can't, right?
Yeah, yeah.
Okay. Good.
It's having, yeah, I got it.
Okay, okay,
Yeah.
wonderful. So let me go to the next slide. So 2 years ago we met and it was a wonderful time. I had such a good time meeting you all in person. And just a few days I took a picture how the t-shirt looks now, and the face looks kind of sad, and that's my feeling as well, because I would of course enjoy an on-site conference much more than an online conference. But let me present the topic to you that I want to present, the colistin-resistant Acinetobacter baumannii phage resistance mechanism. At the moment, I'm in Munich, but this is a picture of our campus in China, where I will be returning shortly. But as I said, I'm currently in Munich and enjoying it a lot. It's good weather, and Munich is a wonderful city. So now I have to kind of on the spot delete a few things. Phage-resistant bacteria, antibiotic-resistant bacteria are a major problem. I don't need to tell you about this. And as previously was outlined already, the major pharmaceutical companies have stopped their programs, and developing a new antibiotic is estimated to cost around $1 billion. So it's a very expensive process to search for a working antibiotic. And if an antibiotic would be developed, then it would be kept as a reserve antibiotic. So it would not be a blockbuster. So therefore, antibiotic resistance is a problem. Companies are not producing any antibiotics, and we are thinking that we will suffer from about 10 million deaths per year on multi-drug resistant bacterial infections. And of course, we have phage therapy, an old therapy which works wonderful, has its problems and has been quickly abandoned due to these problems because we had the wonderful antibiotics. But now we are facing this time where we need to reevaluate phage therapy. So what my lab does is to search for phages, among other things. And we usually use sewage water, which is not very nice to work with. And the students always ask me to do the initial sample taking. And what we do after this is to isolate the phages, just as many of the labs that are also currently listening to these talks are doing. And we isolate phages and characterize them. But when you look at phage resistance, this is a major problem. So we have a lot of very promising approaches to use phages, but phage therapy is severely hampered by the effect of very fast emergence of bacteria that are phage resistant. And I now would like to show you a video. I hope it works also like this. And as you know, is a bacterial lawn, and you see the phage plugs that are growing over time. And if you look closer, after a while, after a few hours, 2 days, something like that, you see colonies in these plugs. And these are actually phage-resistant bacteria. And this is not only common in the lab, but of course, it's also very common in trials. So the majority of clinical trials actually report that there are phage resistance bacteria emerging during the trial, and the very famous trial that has been reported in 2017, where a bloodstream infection of Acinetobacter baumannii was recorded, also colistin resistant, even showed within 1 week of treatment resistance to 8 phages. So it's a it's a big problem. And we have possible solutions. So for example, targeting essential proteins as the phage receptors, or use antibiotic phage combinations, or cocktails of free phages. But despite this, there are always problems with phage resistance. So maybe it's very good to understand the emergence of phage resistance and maybe find ways to limit or even block the emergence of resistant bacteria. So my lab, among other things, started looking at multi-drug resistance in Acinetobacter baumannii in using phages for that. And colistin resistance, especially in China, is rapidly increasing, and it's still a last resort antibiotic. So as previously outlined, many multi-drug resistant bacteria still are affected by colistin. They're still colistin sensitive, but in case they are not, you can use, of course, lytic phages to treat these infections. And colistin is a very interesting antibiotic. It's almost like a detergent. It acts on the membrane. It binds to the membrane and disturbs the lipid bilayer and releases then ions, for example, destroy the protomotor force and lead to death of the bacteria. And so colistin acts on the membrane or on the envelope, and phages bind to the membrane or the envelope. And so our questions that we set out 3 years ago was to try to understand whether phage resistance is influenced by colistin resistance or the other way around, whether colistin resistant influences phage resistance somehow. Good. What we did is look at first at a colistin resistant strain that's called XH198. And if you just count the colonies on a plate or on plates that contain increasing amounts of colistin, then you see a decrease. But if you add a phage at the lower MOI, so at a low ratio of phage to bacteria, of course, excuse me, of course the colony count decreases even more because the phage kills the bacteria as well. So our question was, is this this antibiotic synergy, or what is the principle of this antibiotic synergy? So what we looked at is not only counting plugs, so we also isolated individual colonies and determined the MRC of these colonies towards colistin. And then we also looked at whether they are affected by the phage or not. And what we realized is on the panel on the right here, you can see that there are some bacteria that are colistin sensitive still, or sorry, colistin resistant, but they can still be affected by the phage. Sorry for the slides. These are kind of silly now. Let me try to remove this. So we have, actually I wanted to remove this, that we can see that. So we have bacteria that are colistin resistant in yellow, but they can still be lysed by our phage. But when they are resistant to the phage, they become colistin sensitive. So this is not novel. Jeremy Barr, who also gave a talk on this topic in previous times and has a wonderful paper in this year published in Nature Microbiology, reported also that phage resistance leads to resensitization to antibiotics. And so what we see is that the colistin resistance, this would be also an animation, the colistin resistant bacteria can be infected by the phage, but as soon as they are phage resistant colonies, the bacteria become colistin sensitive. And our question was, how does this happen and why does this happen? And what we did is a whole genome approach. So we looked at colonies that are colistin or that are phage resistant, first of all, and then we did whole genome sequencing and looked which genes are affected and which mutations, and whether the mutations are relevant or not. So we came up with a lot of different isolates, and we found a lot of mutations, but we focused on 2, and these are is a gene that is called AMSE, so it's involved in amylovoran biosynthesis, which is probably involved in the capsule generation, and then a protein that is related to LPS, or in case of Acinetobacter baumannii, lipo-oligosaccharide biosynthesis, and we called it LPS-BSP for biosynthesis protein. So what we did first is look at the genes and whether they are affected, and we found that there are, for example, also resistant isolates, in case here R5 has mutation in both genes, and then R1 has in one, and R7, for example, only in the other. And then we created plasmids with the wild type gene and looked whether the wild type gene, if introduced, can allow the strain to be infected again by our phage, so it can complement. And this is indeed the case. So if you introduce a wild type gene, then the phage can be infecting this strain. Of course, if there are 2 mutations at the same time, this does not work. And next, this is also a bit messy now. We have genetically engineered these strains quite a while back already, and looked at the possibility if the phage can infect and inhibit the growth. And if you just look at the parental strain here in black, without the phage, it grows happily. But if you look here at the lower black line, the parental strain with the phage P24 is inhibited in growth and eventually lysis. But when we genetically introduce the single mutation in this parental strain with AMSE or LPS-BSP, then we see that the phage cannot affect the growth at all. But when we then complement with a plasmid, again, the phage can lyse the bacteria. As a next step, we looked at the absorption, and we only looked at, in this case, our resistant isolates, so not the genetically engineered strain. And what was quite interesting to see is that some isolates show abolished binding, which is has been reported before. So the phage cannot bind anymore because the receptor is altered. So there's more phage in the supernatant here in our assay, the red boxes. But when you look at the other ones, which are the majority, is that you can see there's still very efficient absorption of the phage to the surface, although the receptor is, or our putative receptor is mutated. When we look now at the engineered strains compared to the control, where there's no infection, this is a strain that is not affected in principle by this P24, then we have a lot more phages in the supernatant unbound phage compared to the parental strain. If we look at the engineered strain with the AMSE mutation, we then see that the phage cannot bind anymore. But with the LPS-BSP mutation, we can see that the phage can still bind to the surface. So from this one, from this experiment, we can deduce that the AMSE gene product is or produces a receptor for phage binding. And in case of LPS-BSP, it's not required for phage binding, maybe because AMSE is already serving as the receptor. Since this indicates that at least for AMSE, the mutation changes the envelope of the bacteria, we looked at the bacterial surface. So what we did is embed the cells and then do thin sections and look at the electron micrographs of negatively stained cells. And to me, at least, also analyzing a lot of pictures, we could not really tell a difference between the parental strain or the engineered strain with the 2 mutations. So we still see a fuzzy area around the cells, which might be either due to preparation or due that we can say they actually do not exhibit a phenotype in the TEM. And therefore we use scanning electron microscopy, which is a very useful method to see the surface structure. And compared to the parental strain, we can see that our strain with the AMSE mutation looks markedly different. We have this coccoid shape cells compared to the nice elongated cells here. And they also stick together, they lump together and even form these strings. When we complement them with the plasmid again, you can see that they tend to be more elongated again, like the parental strain. In case of the LPS, we do not see a big difference. Next, we did look at more molecular details. So we did the mass spectrometry of the isolated lipids, and LPS. I just need to remove the boxes here as well, and you can see it. LPS leads to, or their parental strain shows a lipid A peak at about 1910 mass over charge, and this is also present in all other strains. So as the engineered strain that is that has this putative LPS biosynthesis protein mutation, we see a little bit of a different spectrum more in the higher regions, which when complemented is more pronounced. So there's more produced compared to the parental strain. And although we don't know what has changed exactly, we can say that there is a change in lipid composition due to mutation. And in case of capsules, I would point out this was only done with the, this was done isolating the capsule material and then running it on a gel, separating by size. And what you could see is that in case of the parental strain or the LPS biosynthesis mutant, as well as the complemented, we see material appearing on the gel. But when we look at the engineered strain with the AMSE mutation, this is gone, or maybe very little material is visible, indicating that actually the capsule formation is very strongly inhibited by this mutation. When we then complemented with the plasmid, we introduced the plasmid with the wild type again, and we can restore the formation of this capsule material in this mutant. And previously it has been reported that lipid mutants or capsule mutants show attenuated virulence. So we were asking ourselves, is this the case? And what we did is look at Galleria melonella, sorry, so insect larvae, as an in vivo model where we just looked at different concentrations of bacteria. And what you can see is that the AMSE mutant is definitely the least virulent in among the control and the LPS one. And when you complement it, this is the green line here with the plasmid, with the wild type, so leading possibly to higher expression of the gene and the protein or the material that is then produced by the protein, you can see that this increases virulence dramatically, so leads to the death of the larvae very rapidly. With LPS, it's not as pronounced, but the LPS mutant also shows decreased virulence, whereas when you complement it, it increases the virulence again. So back to our synergy, we did the synergy experiment, phage colistin synergy again with all the different concentrations of antibiotic and also at different ratios of bacterial cells to phage. And you see, of course, that the more phage you add, the smaller the apparent colistin MIC is. But synergy would imply that they are really working together. And the question is, are they really working together? So what we did as a detailed experiment, we did that on a single colony level, looking at the colistin resistance of phage-resistant isolates. Of course, we did first the control, where we incubate the bacteria in the absence of colistin and then plate them on colistin plates with different concentrations. And this is done in the absence of the phage as a control experiment. You can still see that there are less and less colonies the higher the concentration is. But when you repeat that experiment in the presence of phage, then first of all, the number is rapidly decreasing, and the levels of antibiotic resistance are also very different compared to the wild type. So when we looked at the detail of our whole genome sequenced bacteria, and I want you to focus on this area, these are all the ones that contain the AMSE mutation and potentially also additional mutations. That's why it's not all homogeneous. When you look at the empty circles, these are the resistant, phage-resistant bacterial isolates, and we determined the colistin resistance, and this is lower than the wild type. The wild type is here at 64, and this would be 8 times less in this case here, for example, at 8. And as soon as we introduce the plasmid again, it goes back to 64 or more colistin resistance. So repeating this experiment with our reconstructed mutant, with our engineered mutant, where we really just mutate the AMSE gene, we see that the resistance drops, and as soon as we complement, the resistance goes back to normal. So really showing and demonstrating that the resistance, mechanism against phage resistance really directly affects the resistance to colistin. Repeating this experiment with our LPS BSP mutations initially looked very promising. There are some additional mutations in these genes, and we saw that the colistin resistance level dropped to 2, whereas when we complemented, it went all the way up again, which made my PhD student very happy. But when we used the reconstructed mutant, it actually leads to a truncated protein, and in this case, it's actually a very, very strong deletion. So it makes a very small truncated protein, whereas this one makes a longer truncated protein because we saw more than one mutation. We actually saw that this is not affected. So we're still investigating the mechanism, whether we need a whole gene and a whole gene product protein for the resistance to be reduced, or if the mutation has to affect the entire gene. And with that, I'm already at the end. And as the summary, I just want to let you know that we also have done a detailed P24 characterization. But what we can say from this work is that there are 2 receptors that are important for phage binding. One is AMSE, so the capsule, and the other one is LPS-BSP, whether the proteins are directly direct receptors, the gene products, or whether they actually just catalyze and produce the receptors is currently not known. The latter case is more likely. And the mutations actually decrease the virulence due to altered envelope structure. And then, as I showed you just now, AMSE mutations decrease the colistin resistance. And maybe we can exploit this discovery of this phage receptors to develop compounds that actually inhibit these targets, although they are not essential for the cell. They decrease virulence and decrease or increase antibiotic susceptibility. So it might be a thought to maybe think about as of these proteins as potential new targets for drug discovery. Okay, with that, I'm at the end. Just would like to thank my group and a lot of undergrad students involved in discovery, but mainly Lydia worked on this work almost on her own. So I'm very proud of the students' hard labor. And with that, I'll open to questions.
Thank you, Dr. Sebastian, for delivering such an informative talk and expanding our knowledge on phage-resistant mechanisms in colistin-resistant bacteria, which is prudent to take phage therapy closer to clinical success. The forum is now open for any questions and discussions from our chair and our delegates.
Sebastian, Dr. Vinod here. It's very quite interesting to know that the phage resistance bacteria showed increased sensitivity to colistin. What could be the mechanism for that?
So I think, yeah, sorry. I think the idea of showing that the capsule is absent in the AMSE mutants is an indication that maybe the colistin molecules themselves have an easier time to reach the surface. So it's possibly just that the capsule kind of prevents the molecules to diffuse to the surface where the colistin would bind. However, saying that in the strain that is not colistin resistant, the capsule formation is not is still there, and the colistin has no problem finding its target there. So it's currently not quite known why this is, but I think it has something to do with the capsule and the absence of the capsule in case of the mutation.
Your finding has opened a new avenue for a research student to, especially to work on Klebsiella pneumonia, which are capsulated, and even they are resistant. So there is a possibility, according to your conclusion, that don't get disheartened if you find phage resistance. So kindly you redo your sensitivity profile so that the drugs which were resistant, could be less resistant, in case of the phage resistance bacteria. It's a wonderful finding. Thank you, Sebastian. Excellent presentation.
Thank you so much.
Thank you, Dr. Lapin. It was excellent work and excellent presentation. Thank you so much.
Thank you so much.
Thank you very much for your presentation. I have a question for you. It's about the phage-resistant clones. This is a stable phenotype, or maybe the phage-resistant clones revert to sensitivity fast?
So once we have a phage-resistant isolate, they, if we even, 1 second, sorry. No, my phone is ringing. Even if we incubate them for extended durations, we haven't done like a long-term evolution experiment, but even if we keep them for like several inoculation series, like maybe for a week, we tried that, we don't get phage sensitivity, we don't regain that. So we have actually a second phage, which we used as a kind of as a stage that only then infects that phage resistant. It's a little bit complicated. So we have a second phage that infects only the first phage resistant strain. So the P24 resistant isolates can be infected by the second phage. And we don't see any reversion of the phage resistant strains that they become sensitive to the phage again. We have not observed that so far.
Therefore, the colistin sensitivity is also very stable.
The colistin sensitivity, yes. So we have not done the experiment that we keep it for long and then see whether in presence of colistin there probably would be an evolution to just spontaneous mutations that would again allow colistin resistance, but I believe that actually these isolates remain colistin sensitive and also less virulent, as we showed in the vivo model.
Thank you very much. Nice work. Congratulations.
Thank you so much. Thank you. Thank you.
Hi, Leptin. I have a question. Great presentation.
Thank you. Thank you, Owen.
Yeah. So the bacteria isolates that you picked up from the plaque, which showed them to be resistant to phage. So you found 2 key genes to have undergone mutation, right? When you did whole genome sequencing. So this is an Acinetobacter. So do you think that there would be, I mean, because both happen to be receptors for phages, in spite of one of them not being essential for the survival of bacteria, but now you think it to be a good drug target, because if you eliminate that, it can make it more sensitive. So my question is that if you work on another phage system, bacteria phage system, so do you think there are some marker genes which need to be studied instead of the whole genome to find out whether that bacteria has become resistant to the phage?
So I think the work that was published by Jeremy Barr is kind of a milestone study, and if you compare that to our work is a little bit late. I think we would have had a very high impact factor paper if we wouldn't have seen the paper by Jeremy Barr. But I have to say, it's wonderful work he has done, and he pointed out that the glycosyl transferases are probably key. And possibly the major phage resistance genes. So, and I have to agree in that sense that because they are all concerning the membrane surface, and I believe that the fast resistance of bacteria to phage is actually always caused by a change of the membrane of the surface structure, whether it's capsule or lipid or whatever not. And I think we always need to focus on the surface genes or that are involved in the biosynthesis of the surface. And that's really key. And that was a wonderful work that was published by Jeremy Barr, and our work complements that work definitely by showing that colistin not only that resensitization to colistin is not only because the bacteria lose the resistance, because they don't definitely they don't lose the resistance mechanism, but that the phage resistance genes directly influence the colistin resistance. That's the novelty of our work.
Right. Thank you so much.
There's one more question, Dr. Sebastian. There's a question from the delegate. She wishes to ask, are there any other resistance mechanisms known in case of Klebsiella phages apart from preventing absorption?
I think I don't know the detail for Klebsiella now, but I mean that general principle is that the bacteria can prevent infection and replication of phages or viruses by a multitude of mechanisms, and absorption is just one of them. There's for example, the restriction digestion, superinfection prevention, even CRISPR-Cas and all these mechanisms. And I think absorption is just one of them. But from my perspective, it's probably the most important during phage therapy, because a single point mutation can lead to resistance, and single point mutations are very rapid. And whereas, for example, if you need to build a super exclusion mechanism or restriction enzymes that digest every single phage DNA, that's a much slower process in evolution. And therefore, I think there are a multitude of mechanisms, but absorption is probably the most important.
Thank you, Sebastian. We are running out of time. Thank you. If anyone has questions, we'll share the email ID of the Sebastian, so you can be in touch with them. Thank you, Sebastian, for accepting our invitation.
Thank you,
Thank you so much.
Thank you.
Thank you. Thank you.
Thank you. Okay, so moving to the next speaker, it's a delight that we have with us Dr. Pilar Garcia. Dr. Pilar Garcia is the head of Department of Technology and Biotechnology of Dairy Products at IPLA CSIC, Spain. Her research is focused on exploitation of bacteriophages as tools to be applied in food industry, including isolation and morphological characterization, sequencing, genome analysis, transcriptomic and proteomic analysis of genes, expression and purification of proteins. At the moment, the removal of biofilms by fast-derived antimicrobials is one of her main interests. And we are delighted to have you, ma'am. Over to you. She would be speaking on fast-derived tools to fight against staphylococcal biofilms.
Thank you very much for your kind introduction. And first of all, I would like to thank Dr. Kumar and rest of organizers for the invitation to participate in this meeting. In this talk, I'm going to summarize the last work from my group about phages and phage proteins to fight against staphylococcal biofilms. Staph aureus is an important threat for human health due to the increase in antibiotic resistance, mainly methicillin resistance. Recent data from the World Health Organization showed that 20% of all infections are caused by EMSA strains, and even in some cases, the percentage can reach the 80%. In addition, Staph aureus is the cause of foodborne diseases. For instance, 7.5% of all outbreaks at the European Union are caused by this bacterium. In addition, EMSA strains are detected in farm animals and also in meat and other derived products. And also, animals are an important reservoir for this bacterium. In the last years, our group has isolated and characterized a small collection of bacteriophages infecting Staph aureus and Staphylococcus epidermidis. Here you can see some of them. There are some images to my electron microscope, and some of them belong to Syphoviridae family and others to Myoviridae family. In these phages, we have identified and characterized several endolysins with lytic activity against Staphylococcus strains. All of them show a modular structure typical from staphylococcal endolysins. Taking advantage of the modular structure of these endolysins, we have obtained new chimeric proteins by shuffling different catalytic and binding domains. This is an example of some chimeric proteins that were obtained from the parental endolysins. And additionally, we have a collection of about 1,000 different proteins that were obtained by shuffling of the different domains and also by binding to cationic peptides. Both bacteriophages and phage-lytic proteins have a great potential to be used as an alternative to antibiotics and biocides to fight against Staphylococcus aureus in different environments. For instance, Staph aureus is a major pathogen associated to food outbreaks. Therefore, new strategies in biopreservation are ongoing. Human therapy and veterinary are also wide areas for application of faith-based antimicrobials against Staph aureus, and interesting research is being doing in prophylaxis and treatment using these antimicrobials. Beyond the specific application of phages and phage proteins as biopreservatives and antimicrobial therapeutics, there is a common need in all these areas for the improvement of the disinfection processes. Removal of Staph aureus biofilms is an important challenge in both clinical and food environments, as biofilms are the main barrier for the action of antibiotics and disinfectants. Staph aureus is a bacterium with a good ability to attach biotic and abiotic surfaces and to form biofilms. Here you can see an electronic microscope imaging of staphylococcal biofilm. Bacteriophages and phage-lytic proteins have been proposed as an alternative to disinfectants for removing biofilms. Indeed, a strategy to efficiently remove biofilms is to target each component of this complex structure, including bacteria and persister cells, but also extracellular matrix composed by polysaccharides and also extracellular DNA and proteins. Dormant cells or persister cells are especially refractory to antimicrobials due to their low metabolic activity, and they are the cause of reinfections. Also, the extracellular material prevents the diffusion of antimicrobials inside the biofilm structure and delay reaching the target cells. Initially, we approached the removal of Staph aureus biofilms by using specific bacteriophages. We tested two phages isolated and characterized in our lab to infect Staph aureus and Staph epidermidis, and these phages were used individually and in a mixture against biofilms formed by 1 and 2 species. Our results showed that there is a clear reduction in biomass formed by Staph aureus biofilms in both cases when phages were used individually and in a mixture. Also for Staph epidermidis biofilms, the treatment with phages removed the biomass and was more effective when a mixture of phages was used. And finally, when we have a dual biofilm with a mixture of the two species, we observe that the mixture of both phages was more effective for reduction of total biomass. But in nature, biofilms are very complex and have a very complex structure. Therefore, we aim to evaluate the effectiveness of phages in dual biofilms formed by only one species sensitive to the phage, and the other species that was insensitive to the phage, not sensitive to the phage. We use Bacteriophage v. Ipla Rodi, and we apply this bacteriophage for treatment of biofilms formed by Staph aureus and Enterococcus faecium, and also against biofilms formed by Staph aureus and Lactobacillus plantarum. Treatment with the phage showed a different result depending on the accompanying bacteria. Thus, biofilms formed by Staph aureus and Enterococcus showed a reduction in Staphylococcus aureus cells, whereas Enterococcus cells remain similar. However, for biofilms formed by Staph aureus and Lactobacillus plantarum, treatment with the phage reduced the total biomass and the viable counts in Staph aureus, but this has an effect in the increase of Lactobacillus plantarum cells. A close analysis was obtained by confocal microscopy. Here you can see samples that were stained with Cyto9 and alive cells were shown in green. In addition, we also stain the extracellular polysaccharide in blue. After phage treatment, a reduction in the number of staphylococcal cells was observed, but in the biofilm formed by Staph aureus and Lactobacillus plantarum, we observe an increase in the extracellular material and also in the number of cells of Lactobacillus plantarum. Here we can see a three-dimensional structure of these biofilms, and we see that the biofilms formed by Staph aureus and Enterococcus were mostly removed by the phage, but biofilms formed by Staph aureus and Lactobacillus plantarum, the biofilms have a different structure. The biofilm was thicker and the phage treatment did not remove all bacteria. The complex interaction of bacteriophages and bacteria in the biofilm structure led us to study some factors that could influence in the efficacy of phages to remove this structure. One of them is the ability of the phages to go through the biofilm. To study the ability of phages to move inside the biofilms, we growed biofilms in a membrane here, and then we applied a phage suspension, recovering the flow through. We study biofilms formed by staphylococcal strains sensitive to the phage, but also two strains not sensitive to the phage, Lactobacillus plantarum and Staphylococcus epidermidis. We observed that after application of the phage suspension at the flow through, there is a reduction in the phage titer comparing with the initial titer for most of the strains. Mainly, this occurs mainly in those biofilms formed by strains not sensitive to the phage, like Lactobacillus plantarum and Staphylococcus epidermidis. On contrary, for two strains, the number of phages increased comparing with the initial value due to the phage propagation inside of the biofilm. These strains are the most sensitive strains to the phage. We have also studied the diffusion rate of the phages in the phages in biofilms formed by different bacteria. These biofilms were composed by two species, one sensitive to the phage and the other not sensitive to the phage. And the ability to form biofilms and the sensitivity to the phage was different in each strain. Then we analyzed the flow through that you can see here, that there was a reduction in the phage titer for most of the biofilms, but there was an increase in the biofilm, in the flow through, in the titer of the flow through, in the biofilm formed by the most sensitive strain. Indeed, the strain sensitivity is one factor that influences the amount of phage that go through the biofilm, but we also detect that there are others, such the amount of attached biomass, or the interaction between the phage and the extracellular matrix. So these results show us that there is a great complexity in the interaction between bacteriophage and the biofilms. Related with this, there is an important question, that is the diffusion of bacteriophages through the biofilm and their concentration at the different parts of this structure. Actually, it is possible that a gradient of phages will be established. Therefore, some areas inside the biofilm are likely exposed to a low phage concentration. So in order to explore the response of cells to a low concentration of bacteriophage, we grow Staphylococcus biofilms in the presence of sub-inhibitory concentration of the phage. Here we can see that for a low multiplicity of infection, there was an important increase in the biomass. This increase in the biofilm formation when the phage is present in a low concentration can also be observed at macroscopic level when the biofilms were formed on polystyrene plates and stained with crystal violet. So we analyzed this by confocal microscopy, and we can see an increase in biomass, in the total biomass, due to a release in the extracellular DNA that you can see here stained in red. We observe that the biofilms form and the phage pressure are thicker and have a greater DNA content. And you can see here, most of these biofilms were stained in red because the stain have the ability to bind to the DNA. We study these biofilms by transcriptional analysis using RNA-seq, and this revealed that there is an activation of the stringent response in these biofilms that were grown under phage pressure. Thus, some genes were upregulated. You can see here that, for instance, it was upregulated the autolysin A, also biofilm-related genes that ICA and RSH. Overall, we deduced that this could slow down the advance of the bacteriophage within the biofilm, so the result would be the phage and remain sensitive to the phage like a reservoir for phage infection. A second strategy to remove staphylococcal biofilms is to use endolysins of phage-lytic proteins. Initially, we tested the activity of endolysin H5 that was previously characterized in our laboratory, and we used this protein against monospecies biofilms formed by Staph aureus and Staph epidermidis. And you can see here in these panels the images obtained by a cryoscanning microscopy. At these right panels, you can see that most of viable cells were local biofilms after treatment with the endolysin. However, there are some cells, and most of the extracellular material that you can see mainly in this, you can see here the extracellular material that remain attached to the surface after endolysin treatment. Another important question is if the endolysins are able to remove persistent bacteria. Yeah, as these are the main cause of regrowth in bacterial biofilms. Excuse me?
Participants, can you mute yourself? Dear participant, I'm sorry for the inconvenience. Pilar, you can continue now.
Can I follow, please?
Please carry on. Please carry on. Yeah.
Okay, thanks. To explore the ability to endolysins to lyse persistent bacteria, we selected persistent bacteria from Staph aureus culture by using two antibiotics, rifampicin and ciprofloxacin, and then these persistent bacteria we treated with endolysin. And we can see here that endolysin was able to remove all these bacteria, that no viable cells were recovered. The result highlights the lytic activity of endolysins as antibiofilm agents and their advantage comparing with antibiotics. Another important question is to know if the use of endolysins against biofilms and the adaptive response of the staphylococcal cells when they are exposed to a low concentration of these proteins. And we study the structure of the biofilms grow in the presence of some inhibitory concentrations of the chimeric protein ChAP-SH3B, that is a chimeric protein derived from an endolysin. You can see here the image of confocal microscopy showing that in the presence of the protein, there is a disorganized structure in biofilms, in these biofilms, and you can see that how the bacteria here are actually located in aggregates. This also can be observed at macroscopic level. You can see here when we grow the biofilms in the presence of proteins, you can see the aggregates. Additionally, we have performed a transcriptional analysis of two strains in the presence of inhibitory concentration of the endolysin, and we also observe a downregulation in the autolytic activity, mainly in the gene A, autolysin gene A, and which is also involved in the formation of the biofilms. Following our strategy to target several components of the biofilm, we assessed to remove the polysaccharide extracellular material, which is expected to release bacteria inside the biofilm and making the cells more sensitive to antimicrobials. In this context, we have identified an isopolysaccharide depolymerase protein encoded by Staph epidermidis phage, DP-OP7. This protein is able to remove the extracellular capsule of the cells. As you can see here in a microscope imaging, you can see how the protein was able to remove most of the extracellular material. And we also confirmed this activity against biofilms. And you can see here how the treatment with the protein removed some of the attached cells in biofilms formed by Staph aureus and Staph epidermidis biofilms. In order to improve these results, we also decide to combine two of these antimicrobials, endolysins and bacteriophages. And we were looking for a synergistic effect against biofilms. So you can see here in this graph the action of endolysin and phages used individually and in a mixture. As you can see, the endolysin was able to reduce the number of cells very quickly, but after 24 hours, there was a regrowth in the biofilm. The bacteriophages, is the white bars, was also able to remove the biofilms, but a little bit slower. But the combination of both antimicrobials was very effective and also were much more stable on the time. We also can see this result using confocal microscopy. You can see here the control samples and those that were treated with the endolysin and with the phage and with the mixture of both components. And we observed that most of the bacteria were removed and the green color that correspond to allied cells were removed, and most of the cells were stained in red because they are dead. Finally, the quantification of the antibiofilm activities we think is also very relevant in order to compare the different proteins and also to find the optimal conditions for biofilm removal. So we have used a real-time cell analyzer to measure the biofilm formation and dispersion. The attachment and growth of the bacteria to the wall surface modify the impedance values here. So we can use this technology to measure the activity of our proteins. Here is an example of the antibiofilm activity measured for several phage proteins against Staphylococcus aureus biofilms. We observe a decrease in the impedance value for both endolysins and chimeric proteins, and also we observe a decrease in impedance for the biofilms that were treated with isopolysaccharide depolymerase. So my conclusions are that bacteriophages and phage-lytic proteins and also isopolysaccharide depolymerases have a potential as antibiofilm agents, but of course, several factors are involved in their efficacy. We showed that inhibitory concentration of phages may induce biofilm formation, but on contrary, inhibitory concentration of phage-lytic proteins inhibit biofilm formation. Overall, new studies are necessary to improve biofilm removal by combining these antimicrobials. And thank you very much for your attention.
Thank you so much, Dr. Garcia, for the very useful insight into the role of phages as potent antibiofilm agents. The session is now open for any questions from our chair and our delegates.
Dr. Pyler, it was excellent work and a nice presentation. Thank you so much. Audience, if you have any questions, you can ask her.
Thank you.
Actually, I have a question. In one of your experiments, you tried lysine, endolysine, and phage separately, and you have shown that lysine was better than phage, right?
Yeah.
If I understood that. Yeah. So I was wondering, for the biofilm activity, the activity of phages on biofilm is because of killing the bacteria or disintegration of the other components of biofilm. What I mean, killing bacteria is enough for disintegration of biofilm?
I suppose that that no, because we need to remove also the extracellular material, of course, because if this material is attached to the surface, it is also there is a possibility that other bacteria attach in this part. We observed that the bacteriophages are more stable on the time. I mean that, for instance, when we apply endolysine to a biofilm, this is very, very effective, but the endolysine is inactivated in a short period of time. So endolysine is very, very has a very quick activity, but in a short time, the protein is inactivated.
Because of the
This is because, yeah, this is because we have combined with bacteriophages, and we are also planning to combine with the isopolysaccharide depolymerase, because we need to remove most of the extracellular material to make the cells more sensitive to phages or to endolysins or whatever.
Did you try to see whether it remains alive bacteria after phage application?
It remains live bacteria?
Yeah.
Yeah, yes, of course. There is some bacteria that are inside the biofilm that are, for instance, persisters or dormant cells that are not sensitive to the phage. So then there are dormant, but you can recover these alive cells. Yeah.
Thank you for a nice presentation.
Thank you so much.
Thank you. Great presentation, Peter. I have a question. This phage, as like you would have tested these phages first against the bacteria, planktonic bacteria, before testing them on biofilm. Is that right?
Yeah, of course. We tried the activity of phages against both planktonic and attached cells.
So did you also try to add the phages once the biofilm was formed, either phage or lysine or combination? Or did you just infect the phage and bacteria together and looked at the impact?
We use the phages to treat preform biofilms, and also to add to the cells before the biofilm was growth. So, for instance, if there is a clear relationship between the age of the biofilm and the ability of phages to remove them. I mean, for instance, for very mature biofilms, there is more difficult to remove these biofilms by bacteriophages and also by endolysins, but even mainly for bacteriophages, because in these mature biofilms, most of the cells are in a stationary phase. So there are like a dormant cells, and bacteriophages prefer to infect active, metabolic active bacteria.
Right. So that way, phages may not be disrupting the preformed biofilm very much, right? Lysins could do that better. Because
Excuse me?
Now, as you said, that because once the biofilm has formed, which means cells are now in the stationary phase, and therefore phages may not be able to disrupt it as well as lysins do.
Yeah.
But the lysins are not stable for long incubation times.
Yeah, are very quickly, are even more effective than bacteriophages, also against stationary cells, but the stability of the proteins, it seems that is very low, that the stability of the phages. So we propose to combine both because it's a way to remove biofilm during a long time.
Right. Thank you so much.
Thank you.
Dr. Garcia, I wanted to ask one thing. This is Amita Gupta from Delhi University. Have you tried a combination of phages and lysine molecules to disrupt preformed biofilms?
No. No, we use individually biofilms grown in the presence of bacteriophages, as I show you, and also endolysins, but not a mixture of both. It would be a good idea.
Do you think that could be helpful in getting the phages to the bacteria properly and then allowing them to enter into the bacteria?
Could you repeat, please?
I'm saying, do you think a combination could be a better agent than trying only phages for the biofilms?
I think so. I think that the combination would be better than individually use bacteriophages or endolysins. I think that is a good idea.
Okay, thank you.
Thank you to you. Welcome.
Dr. Ramesh and Dr. Sanjay Chipper wants to come in. I think he's not able to join.
Sorry?
Dr. Chipper is trying to join in, but I think
okay.
Let him in.
Yeah, I'll take that. Sandeep, you are on mute. Sandeep, you are on mute.
There is one question from the audience. I think we'll take up this last question. Endolysin used in the study is encoded by the same phage that you were working with or from other phage? This is question from Mansoora, Miss Mansoora.
It's from the same phage, but it could be from another one. I think that there is no problem with this.
Okay. I think Ramesh, sir, we can.
Thank you,
Yes.
Paila, for excellent presentation and for accepting our invitation, to be the key speaker, for this conference. And almost your work, has a lot of similarity with my work, and definitely I'll be in touch with you, ma'am. Thank you for an excellent presentation and for your time. Thank you very much, ma'am.
Thank you. You're welcome.
Thank you,
Thank
Dr.
you
Pillai.
very much.
Thank you.
Thank you very much.
Sanjay Chipper has already joined. If he has any question, he can ask.
Yeah.
Sanjay sir, are you there? I think he.
Okay, thank you. Thank you. Thank you very much.
Okay.
You can go for the next speaker. Sanjay.
Yes, sir. Yes. Sir. So we're moving on to the next speaker. We have with us Dr. Bülent Bozdogan. He is presently the Senior Professor, Faculty of Medicine, Department of Basic Medical Sciences, Aydın Adnan Menderes University, Turkey. Dr. Bozdogan did his MD degree in Ankara, Turkey, and PhD degree in Paris, France. He then studied on antimicrobial resistance mechanisms in gram-positive bacteria, and following his postdoctoral studies on molecular epidemiology, he further studied in Pasteur Institute as Deputy Director of Antibiotic Resistance Reference Center. Since 2005, he is working at Adnan Menderes University, Turkey, and is the Director of Recombinant Protein Research Center. He has also studied in VIT India in Dr. Ramesh Sir's laboratory on antimicrobial resistance and phages. We welcome you, sir, and he will be enlightening us on the topic genetic engineering of bacteriophages. Over to you, sir.
Welcome, Dr. Boland, sir.
Thank you very much. I cannot share my screen because there is another presentation still going on.
Yeah. Dr. Pilar, can you answer your PPT, please? Dr. Pilar, are you on the line? Dr. Sandeep, can you call her? She has to answer. Or the admins, can you remove the PPTs? Admins?
She's there.
Dr. Pilar, can you answer your screen?
Dr. Pilar, ma'am, can you please hear us? Hello?
I think admin can remove the PPT. Admin? Dr. Vinod, sir?
They are trying. Give some time.
Yeah.
Please.
Hojam, sorry for the inconvenience.
They're working on it. They're working on it.
Yeah.
I don't know how to proceed now.
Yeah, please, can you answer your screen?
And
Can
now?
you stop the sharing? Can you stop the sharing, please? Yeah, good. Thank you.
Thank you. Thank you, ma'am.
Thank you.
Yeah.
Thank you. No. Yeah. You can see my screen now?
Yes, Hojam, you can.
Okay.
So, okay, everything is perfect.
First of all, thank you very much for invitation. I'm very happy to be there. Even I would like to be there in India, but it's a way to be there, I guess. So I would like to thank to the organization for inviting me, but also to Dr. Ramesh, because a couple of years ago, 2014, I spent 8 months in his lab, where I learned phage studies, where I started. For this reason, I would like to thank especially for Dr. Ramesh.
Thank you, Hojam.
So today I will talk about genetic engineering of bacteriophage. It's a very big title for my small talk. First of all, I should say that it's not, surely it will not cover all over what is done for genetic engineering of bacteriophage. I will show some examples on the matter. So, it is already said a lot of times, bacteriophage are one of the main actors of biological mass, they say. The numbers change from one publication from another. More than 10 to 30, 32 phages are existing. So the researchers consider that these numbers should be at least 10 times higher than the number of bacteria. So also it's said that half of the bacteria existing are killed by phages, especially in ocean. which adds a lot for evolution of biosystem. So if you think about our intestinal flora, in the morning, one of the talk was about this one. In our intestine, we have colon. We have, for number, much more bacteria than our cells in total. So if you think about phages, 10 times more at least. So we have a lot of phages inside us also. So they are everywhere. So why do we need genetic engineering for bacteriophages? We can resume in two titles, I guess, to obtain better phages, especially for bactericidal activity. The natural phages, surely they are very effective, but they do not cover all of the bacteria existing isolates. For example, in one of our study about Pseudomonas aeruginosa, which were isolated from diabetic foot infections, we tested 10 phages that we have isolated. Even in combination, we couldn't get more than 89% of activity against the strain. So if we want to have 100% activity, we should do something genetically probable. The other thing, phages are not used only for the treatment purposes, also some other functions. For example, they are very good markers to show presence of bacteria. Even in low number, we can obtain high number of phages, so we can show their presence. If we change the phages by a marker detectable, like fluorescence or luminescence, even better we can detect the presence of these bacteria rare. Also, another tool, the bacteriophages used as a tool for phage display. If we put on the surface of the phages an antigen, and it can be used for vaccine purposes, they say. So what we have as problem for the natural ones, why we are looking for other solutions. One of the limits of the problem with the phage therapy of natural ones, limited host range. Sometimes it's very good to have specifically target one species, but not the others, especially for protecting the floral bacteria. But the large host range is sometimes needed, and we can, to redirect the phages for another species, we should use some genetic formulations. So in a lot of talks, it is cited that some bacteria are resistant for the phages, and to overcome this, also we can use these genetic methods, synthetic biology methods, especially. Also, one of the problems with phage therapy for the gram-negative bacteria, if we kill them rapidly and disintegrate them, we will have some free lipopolysaccharide, which is a component of their outer membrane. So for which immune system is activated. So toxic shock syndrome develop. So surely even with the antibiotic treatment, we should avoid this one. For this one also I have one example of study. Also, the phages was found in India first from Ganga River against, if I remember well, cholera bacteria, and then some others developed and find, especially doctor, he wasn't a doctor, Darrell, researcher, he used for therapy against infections. So, but it was very difficult to send phages from one site to another site, like a commercial antibiotic. So stability problem was one of the important problem at this stage. So after development of antibiotics, it's said already in 20th century in the half 1950s. So phages are forgotten, as Dr. Kumar said. So what we can done, on which we can play for to make the phages better. One is playing with the receptor binding site, which is usually tail of the phages. I will give one example about this one. Or reduce endotoxin release activity. Also, which is important if we apply by intravenous way, IV way, the stability in blood is very important. So longer circulation is better activity, surely. For this, also there are some researches. I will talk about these ones. So one of the studies done for host range expansion is this one. It is published in 2002. This is a polyphage, FT phage, which is used for phage display studies. It has especially two proteins on the surface, major and minor proteins, P8 and P3. This phage infects especially E. coli. So researchers found another phage which has a similar P3 minor coat protein, and some part of this phage protein, gene coding for this protein, amplified and cloned. So combination of achimeric one, combination of these both P3 from E. coli and P3 from Vibrio cholera phages effectively infected and released both E. coli and Vibrio cholerae. It's one of the examples for this study. Even they got a patent for this one. The study was done with Massachusetts Institute of Technology, and what they have patented is recloning and reassembling part of phages in yeast artificial chromosome. They got patent for this one, and it works well. So the second problem is whenever we apply phages intravenously, IV, the immune system produces some antibodies or cellular immunity develops against these phages. So this is a recent study published in 2019 by Chinese, and they found that some phages could stay a longer time in circulation than other phages. And they found some motifs in these blood circulating prolonging peptides. And one of the motifs, which is combined arginine, glycine, and aspartic acid, is the important one. Is repetition of these motifs allow phage to hide itself from immune system. So because this motif can able to bind to ferritin, human heavy chain ferritin, and so it can be avoided phage exposure to the immune system. So I don't know if you can see here, there are a couple of figures from this study. This is at 0 hour, all the phages, and after 72 hours, this is the control one. These are the blood circulation prolonging peptide phages with these peptides, especially BCP1, which has this RGD motif. So the number of phages in blood, whenever it is BCP, it remains always high up to 144 hours, which is decreased after 48 hours for the control ones without BCP1. So it allows this presence of this peptide if we genetically change the surface protein with these peptides, it can stay a little longer. They didn't do this study to give a little more life to the phages. They did this study to be able to fabricate some capsules to put their anti-cancer protein inside. So what they did a capsule with this one. They put their anti-cancer agent, and so it stayed prolonged blood. It had prolonged blood circulation, and also the uptake of this anti-cancer agent was increased. So bacteriophages are used also for diagnostic purposes. I mentioned a little in the beginning. So one of the ways is including in phage genome some reporters like fluorescent or light reporters, luciferase. So if they are included in the genome of the phage, so we can detect the increase in light or fluorescent in the sample. So we can show presence of bacteria which are infected by these phages. So it is one of the important way. It is the only way for the genetic engineering method can be used. The others are, for example, by phage itself, giving the phage, a natural phage, and to see the proliferation of these phages by classic agar method. And we can see that, there are some bacteria, even low numbers, by counting the plaque formations. The other is some specific gene for these phages can be amplified by qPCR, and then we can show the increase in the number of these amplicons, which shows presence of bacteria. So one of the reporters is done with T7 phage. They included a nanolip reporter, luciferase-like gene. So they amplified genome of the phage by PCR, different fragments, and using Gibson assembly method, they ligate all these fragments with this nanolip reporter gene. After assembling this DNA, they did transformation and they got the phage. So it's so in this study, this is in light, this is in dark. They have taken two pictures of the same plate. You can see the plaque formation here, some plaques, and you can see the light emission can be observed like here. It means presence of these luciferase-like gene in the genome of phage. So if we detect the light luminescence in special units, surely each unit can show presence of bacteria. For example, here it's arbitrary unit 1000 units correspond to approximately 10 to 4 bacteria presence of. So it can be used for detection of bacteria. It is a recent study published in 2019. Another way of using phages for presentation of antigens. So this is the FT phage that I talked about a little. Two proteins, major and minor coat proteins, can be used for phage display studies. In this study, published in 2012, they included an antigen for the major capsid protein. Even they included two different antigens together. So what they done, especially to include for the minor quad protein, they added a part of antibody variable part of antibody against dendritic cell. So it was to target directly the antigen presenting cells, dendritic cells, and to amplify and to make it quickly development of antibody against the antigen presented on the surface of phages. And they said that it worked well. So if we return back to the phage therapy, what can we do to make better a phage for the therapy? We know that there are a lot of unknown genes, unknown possible hypothetical proteins in the genome of phages, more than 50%. We know about 1/3 of the genes, approximately. The others, we don't know the functions yet. So except these ones, we know that some phages carry toxins or virulence factors which harms the host. Host means not the bacteria, the host that bacteria infect us. Also, the presence of antibiotic resistance genes is important, which are carried by the genome of the phages. Except these ones, the endotoxin release that I talked about, which causes toxic shock syndrome, is very important for the survival of the patients. Also, one of the important subject for me, lysogeny genes, to have the lytic genes from lytic phages from lysogenic phages, they should be removed. So in the morning, Dr. John, I guess, he talked about holins and the importance of this one. In this study, published in 2005, by Amber mutation, which means by adding a stop codon in the gene, they caused holin knockout. So they have get rid of holin presence. So after this one, they obtained lysis deficient bacteriophages. Normally, it shouldn't work, but it works, they say. And they did an experimental peritonitis in mice and used for infection E. coli. So this is the number of E. coli after this study. And this is the percentage of survival of mice after treatment. So first of all, let's look at to this graph. If they don't use any phage or any antibiotic, the number which is given 10 to 7 E. coli increased to more than 10 to 11. So this is the control one. And with the treatment, they used two phages and one antibiotic. This antibiotic is a beta-lactam antibiotic. So they used wild type phage, and this is lysis deficient phage also. As you can see here, in 2 hours, the wild type phage killed almost all the bacteria. So it worked well in vitro. Sure. The infection model was peritonitis. It's not in vitro, sorry, it's in vivo, the number of E. coli that they have given. So when they used antibiotic, it started to decrease, and after 4 hours, it was below approximately 10 to 4, which means bactericidal 3-log decrease from the beginning of bacteria, number of bacteria. So 3-log means bactericidal activity. For the lysis deficient bacteria, there was nothing till 6th hour, and after 6th hour, it was shown that the presence of E. coli was like wild type phage. So the other side is the survival. You know, all of the control mice are dead after 20 hours with the infection peritonitis. So whenever they use antibiotic, beta-lactam antibiotic, approximately more than 60% of the mice were dead. It's not because of the anti-E. coli, but because of the toxic shock syndrome. So wild type phage was better than antibiotic, which shows that even for the wild type phage, we can obtain a better result than antibiotic for the endotoxin activity. But whenever they use this lysis deficient bacteria phage, and more than 81% of the phages, mice were alive. So it is a very important result, I think. So these are the level of endotoxins for control and for control after 6 hours and after 12 hours. For the control, it was surely in the beginning it was low, and after, because the bacteria die, remained approximately 50,000 units. But if you check for the antibiotic, it was the highest after 12 hours. The level of endotoxin in the periton of the mice infected and treated, and the followed by wild type phage, and for the lysis deficient bacteria, they got very low, even lower than control level of endotoxin. So another study is, this is published in 2021, and the idea was reduction of the phage genome to get rid of some part of the phage genome that they think that it wasn't necessary. So they amplified, and it's with the same method, the artificial chromosome method, they assembled and transformed the bacteria, they got the phages. You see, the decrease in the was approximately 4 kilobase of part of phage from approximately 40 kilobase were discarded with this method. So the activity was almost the same. So the last part of my talk is about prophages. You know, as we shouldn't be paranoid, but the viruses are everywhere. Even with the COVID, we have seen that. But in us, also also bacteria, they have a virus inside their genome. So when they have sequenced the human genome, they found that from 8 to 14% of human genome was consisted of consist of viruses. So for the phages, it's almost the same. And if you can see here, they have shown for bacteria for the presence of prophages, phages which are integrated in the bacterial genome, and 16% of total genome of E. coli consists of prophages. It was 12% and a little less for
participant, can you mute yourself? Excuse me, Hoja. Participant.
Yeah. So we have an important source, let's say, source of phages inside bacteria itself. So we plan to do a study we started already with Dr. Ansaldi from France to generate minimal virulent phages from prophages in Salmonella. I will make a little bigger this one. Oop, another. Yeah, I don't know if you can see. Yeah, it's better, I guess.
So too bigger, Hoja.
You can see the.
Yes, Hoja.
So. Yeah, I was doing with that presentation now. No, that's too. Yeah. I did that too, I think. Yeah. Let's turn back to the original presentation. So, I couldn't do, sorry about this.
Hojam, nothing, the
technical problem.
Zoomed it, nothing, just reduce
Yeah.
Yeah, now we got it.
I will show like this, even if it's smaller, it can be understood, I guess. Let's say we start with this one, Salmonella genome, presence of two phage. Now it is very easy to find whether in a genome there are some phages or not. So, and it is also easy to induce these phages by some molecules or physical stress. Physical stress, we can do this, we can produce some phages. And if we can isolate these, separate by from agar, and it became very bad. Well, sorry. So the whole idea is to randomly do restriction of these isolated genome from these lysogenic phages and do ligation and do transformation after, and to choose the phage, lytic phage, which has the smaller genome. So the idea was this actually. So I will conclude with this one. Surely we, for the antibiotics, why they became more important than phages in the beginning of 20th century or in the middle, because they could be commercialized, they could be produced and sold. So for the phages in our system, which is based on money and capital, we should have some product to be able to sell them. I think for the future of phages, we should play on these ones and to have some products that can be commercial, and then it should be sold to be able to use by everybody. So that's one of the reasons that synthetic biology is needed, and especially to get rid of, in our study, I tried to show, but it didn't work well. To get rid of these hypothetical proteins or harmful genes, and only get the minimal lytic phages. And it should be, I think, the future of the phage therapy. So I use these references for this presentation, and thank you for listening And this is from Aydın Kuşadası. Yes,
Thank you so much, sir, for the very informative and wonderful talk given on phage engineering. And this is very crucial for the success of phage therapy, that we need to optimize the phages. Now the forum is again open for any discussions and question answers.
Sir, this is Amita Gupta from Delhi University. I wanted to ask you about the E. coli detection assay that you showed with the luciferase phages, in which you showed that the DNA was assembled with the luciferin gene by the Gibson assembly method, and then phages were allowed to infect and check the presence of E. coli.
For which one you are talking? For this one?
No. Earlier.
This one?
For diagnostic purpose. The next one.
For that.
And the one after this. This one.
Yeah, they did sequence. The amplification of genome was by PCR, so they used their special primers to amplify the region that they want to amplify. So, and after they surely sequenced the phage. Yep. It was the question, right?
So they are incorporating the luciferase gene in this phage. How does the phage show fluorescence?
How did they do this phage? Yeah, they the same way, the Gibson assembly way they used. They amplified, they ligate, and after assemblation, they did transformation. Gibson assembly method.
Yeah. So they incorporate the luciferase gene. How do the phages become fluorescent? They are incorporating it in the genome.
It's not fluorescent. It's luminescence.
Okay,
it's
luminescence.
Not very important part.
Yeah.
So they amplified also this gene, and they included it in the genome. So Gibson assembly. You amplify with different, they gave all the sequence of the primer in the study, if you check, and they bring together, and if they came together, so they have the lytic phages.
Yeah.
And this
is.
Thank
you. Thank you.
Yeah. Thank you.
Thank you, Ojam. Thank you so much. Thanks for your time. It was a wonderful presentation. Dr. Sandeep, are you here? Yeah.
Yes, sir. So, okay. Thank you so much, Dr. Bulent, sir, for the informative talk. I think you can stop sharing the screen.
Yeah.
Yeah.
I will stop sharing.
Thank you, Ojam.
Thank you. Stop share, yeah. I found it.
Thank you, sir.
Thanks.
Moving on next to our
Bulent.
speaker. Yes, sir.
Thank you, Bulent, for participating in the conference. It was a great presentation. I think Bulent left because there was a network issue from my side. I totally agree
I'm
with him.
still here.
Bulent, I totally agree with you. The scope of phage therapy lies with a genetic recombination, because I always support the genetic recombination of the lambda virus, which can customize the bacteriophage in the hospital setup. It was an excellent presentation. Thank you, sir. Thank you for accepting our invitation also.
Thank you
sir.
very much for inviting me. Thanks.
Welcome, Dr. Mata for the conference.
Thank you. Nice to
Welcome, ma'am. So we have our speaker, Dr. Martha Clokey, with us. She's a professor of microbiology at the University of Leicester, where she leads a large group of researchers working on therapeutic development of phages that target human and animal pathogens. Her work encompasses all aspects of phage therapy development, from unraveling the fundamental biology to product development and commercialization, ma'am. You have 3 patents filed from your work and numerous international publications to your credit. We welcome you, ma'am, on this forum, and we would now like you to enlighten our delegates with the talk on getting phages closer to clinical deployment. Over to you, ma'am.
Thank you. Well, thank you very much for the kind introduction and for the invitation to be here. I'm sorry I'm not with you in person, but hopefully I can at least share some insights from my laboratory over the last few years. So as was said, the title of my presentation is going to be Getting Phages Closer to Clinical Deployment. And I want to give some examples from recent work in my lab looking at urinary tract infections. Now, much of more information indeed of what I'm going to talk about can be seen on the website that we now have at for our lab, which is the details of which are here. So leicesterphagelab.co.uk. If anyone would like to read more afterwards, feel free. So the structure of my talk, I'm going to touch very briefly on the antimicrobial resistance landscape. And then I really want to spend time in 3 areas. The first is looking at how we can better characterize phages. So we can find phages for many diseases where we would like to use phages, but how do we make sure we have the right ones? What tests do we do? What studies do we do to ensure that all of the long developmental pathways are done with the absolute best phages? So I will give you some share with you some data and some approaches that we've been using successfully. And then I want to talk a little bit about taking these ideas further to actually identify ecological types of phages, so that each time we want to study a new disease, we don't have to start invent the wheel all over again. So I'll share with you some things in that department. And then I will talk about where it's best to use phages. And within that context, I'll talk about our urinary tract infection work. Okay, so that's the 3 parts of the talk. So just to touch on, though, as I said, the antimicrobial resistance landscape. I'm sure other people have shown you this slide, and you all know that if we don't do something about antimicrobial resistance, it's predicted that by 2050, we'll have 10 million people per year dying of antimicrobial resistant infections. And we've just seen, we're in the midst still of this horrible global pandemic. We've seen what happens when we don't do something about well, in this case, about viruses or antibiotic resistant bacteria. Both are extremely important to tackle. And often antimicrobial resistance is not seen as being so urgent, but really it is. And we can see from this map that a vast number of that predicted 10 million are thought to be predicted even to be in Asia. So it's useful to think about, though, before we go into the details of the talk, well, how will we actually use phages? Because depending on how we use them, we'll then develop them differently. So will we be using them simply to treat infections that antibiotics can no longer treat? Or will we be using them as frontline therapies and therefore prevent antimicrobial resistance? Will compassionate cases become more routine? Or will it be more common to do more clinical trials, and thus doctors will be able to access phages through standard routes? So how can we get phages into mainstream medical systems? It's really sort of key to think about and work out how we can get to this point. So the first thing that I'm now going to spend the first part of my talk talking about is, well, in order to get phages into clinical use, we have to make sure we use the very best phages. So how do we do that? This is a picture to make an analogy with botany on plants. So I'd studied a lot of botany before I studied phages, so I've only been working on phages for 2 decades. Before that, my grandparents actually were all botanists, so I was brought up with this culture. And when it comes to botany, if we want to find or identify particular plants, we can do that. We can go to the place where interesting plants grow. So this is in England, actually. This is a chalk grassland, which is very, very species rich. So we can go here, I can open my botany book, because hundreds of botanists have worked on these plants over many years, it's possible for me to identify what plants are surrounding me. And then if I wanted to do something like restore a garden in a particular way, I would know which ones to select. But we can't do that with phages. We can go to environments where we can find phages, such as natural environments, such as here is an example showing a mudflat. But even if we could see the phages, or we can indeed isolate them and look at them, but we don't know which phages have which features, and which phages we want to use will be good ultimately for using therapeutically. And that is because the book has not been written. So people like myself are really interested in trying to identify what kinds of phages are going to be the most effective for us to use. So I heard in the last talk, and I agree, there's definitely going to be a role for engineering phages, but I think there's a vast role to actually figure out how particular types of phages work, and to actually select good natural phages which have good properties. So when it comes to isolating phages, some are easier to isolate than others. So some bacterial species, we can very easily find phages, things like our E. coli, for example, Pseudomonas, but other species are much harder. The first thing we do very often is just look at them. I always love to do this. These are just a whole bunch of phages that we isolated recently in my lab in Leicester. We can see the different morphologies. It just gives us some indication of how they work. It's a good starting point. And then we can look at their other properties. So we can look at how quickly they kill bacteria. So here we have a cleared culture of bacteria compared to the control. And then within that, we can find phages which kill completely and can kill aggressively. So phages, like other viruses, don't immediately want to wipe out their host. That would not be advantageous to them biologically, but some will be more aggressive than others. And in therapeutic for usage, we're probably trying to find ones that kill all of the bacteria. So this phage shown in a kind of, what color is that now? It's this is the blue. If you look at the blue phage, that's killed everything in that culture in 5 hours, whereas other phages haven't killed all the bacteria. So we do essentially what we call killing assays. We then very routinely now go to the genomes of phages to check there's nothing in that genome that's going to make the bacteria worse in any way. So we don't want any toxins in there. We certainly don't want antimicrobial resistance genes. We don't want evidence of lysogeny either. We can also look at the proteomes of the phages, and I'll talk a little bit more about other omics in a minute, but these are some of the basic characterizations we do straight away to try to at least have a starting point on whether we think that phage is interesting. Now, what we then have to consider is, well, how are we going to use that phage? So that phage, actually, it has to be formulated in such a way that it can be stably delivered to the relevant tissue or organ within the human body. And then when it's there, it's got to work in the right kind of microenvironment. So for example, if the phage is expected to work in the lungs, it will have to work in particular conditions of different oxygen concentrations, or if it's under a mucose, under a layer of sticky mucose, it's got to work anaerobically. So we've got to consider how that phage will be needed to work to clear an infection. And then we want to make sure that they're effective with other phages. So in general, if we're using natural phages, we need to use multiple phages to reduce resistance. Now we need to consider, therefore, what phages will work well together. So you also need multiple phages because in general, we need to get over the specificity issue. So in general, if we want to produce a phage product that doctors can easily access to have an off-the-shelf product, for example, that phage will need to cover all of the different variants of that bacteria. So you'll need multiple phages probably to do that. So we need to make sure those phages will work well together. And then we'll need to make sure that they work well amongst other bacteria. So there's no point in just having phages that look good in culture if they don't work for any of these downstream processes. And as I've worked on phages more and developed more things for therapeutic endpoints, I've become better at considering these downstream considerations earlier in my pipelines. So I did recently publish a paper with my lovely postdoc, Janet Nale, where we published all of the different models that are commonly used by us and other people to study phage efficacy. So if people want to read more, that can be seen in this publication here in Nale and Kluki in Current Opinion in Biotechnology. So I'll talk you though briefly through some of these methods. So what we want to do is we want to go from just finding phages that look aggressive, and obviously we will have to, before going into humans, go into animal models to show efficacy and safety. So with Clostridium difficile, I've spent a lot of time working on those phages. C. diff is a problem globally in India and well across the world. Still, it's not a problem that's gone away. It's serious bacterium that causes diarrhea, particularly in hospital settings. So what we can see is in a hamster model, which is what you use to study C. diff, different combinations of phages can reduce significantly the amount of colonization in a hamster gut. Okay, so we can see different combinations work better than others. So this is a very good combination just of those two phages. And we also showed that the phages can double the life expectancy of the hamster. So the hamsters are really sensitive to the toxins that C. diff gives out. So this is very good data which we've been building on. But we don't want to go straight to animals because it's generally quite expensive. There's complicated ethics and they're very time-consuming experiments. So you can do a lot of optimization of your phage sets before you go into those animal models. So I'll talk you through some of those. They are things like looking at biofilms. So this is a C. diff biofilm. And can you see these phages that work brilliantly in biofilms? We can see the patches of clearance in the biofilm. And actually, what's interesting is that in general, if you use antibiotics with phages in a biofilm model, the phages will make the antibiotics work better because they have enzymes that penetrate the biofilm, and that allows also the penetration of the antibiotics. So we do quite a lot of biofilm work because, as I said, most infections are present as a biofilm. We've also done quite a lot of work in epithelial cell lines. So in general, for example, in C. diff or Salmonella, we've done quite a lot of gut infections and also lung infections. You'll be treating that mucosal membrane. And I know there was a nice talk earlier this morning by Jeremy, who's done a lot of work in this area. And we've done also a fair bit, and we've particularly focused on how effective phages would be when you add them to the epithelial cell lines. So this is some work that was headed by Jinyi Shan in my lab. And essentially, I just show you the data here. He showed that if you add phages to bacteria in the context of a cell line, actually they kill much better. That's this graph here. They kill much better. They kill more bacteria than when the C. diff and the phages are growing together. So presumably the epithelial cells are acting to anchor and bring the two partners together. We also showed that phages can replicate very nicely in that environment. This is data showing that they effectively clear a biofilm within human cell epithelial cells. And importantly, that there is no, if you look at the LDH release as an indication of toxicity from broken cells, or something the phages may do, we can see that there is not very much, there's minimal LDH release. So this is all quite positive. So you can do epithelial cell work, I think, very effectively to really try to understand how your phages might be interacting with their cells. And then obviously different types of epithelial cells depending on which clinical indication you want to treat. Now, another brilliant model which we've used lots, actually this started with a wonderful Indian connection, a young Indian scientist who was really interested in using phages to prevent the infection in silk moths. He's called Mahananda Chisia from Assam. And he was supported by the Indian Institute of Biotechnology, and he came to work in my lab, and he wanted to work on these silk moths, but of course they don't grow when you remove them from their natural environment. So we started to use wax moths to try to help him. But then since then, we've realized it's such a good model for many infections and phages that we've used it a lot. So this is showing you how it works for Clostridium difficile. And we can see that with C. diff, we worked out the dosage that if we give the moth larvae, we know that in 60 hours, if we do not do anything about it, the larvae are all dead. This just shows you the kind of colonization on the right side. And we have a very good visual indicator because these are the healthy wax moths. And this is when they're dead, they go black, they go from gradually darker until they die. And this is just showing you that we can easily remove phages from these things with time. So what we showed when we add phages into the system is really fascinating, actually, because if you add phages prophylactically, this is the top line. The top line is both prophylactic use and also the control where you just gave the animals phages and didn't give them any bacteria. And we can see that actually, if you add phages prophylactically, we cannot kill the larvae. Okay, so the phages prophylactically look very, very effective. We can, in a remedial model, we can save a number of the, we can save about 25% of the larvae. That's just with one dose. And we showed actually, I'm not showing you all the data, I'm just wanting to highlight a few different models today. But we showed that we really can bring this, if we add multiple doses, we can get this level of survival back up to about 80%. So they do work well also when used as a treatment, but really well when used prophylactically. Now, we've also got this model working with salmonella. So again, you can see these are the larvae that just had salmonella, they all died, and with different phage combinations, they've done slightly better or worse. So this is actually a really good model also, if you want to do things like look at how virulent your phage-resistant mutants are as well. So we use this model to answer quite a lot of questions. Another model that's really useful is to use an artificial gut model. So this is an example of that. So this is what we do here. It's very simple. We just take fecal samples from a number of donors. So we use 4 different donors to get a really nice diverse set of bacteria. So we use different age profile of different donors. And then what we can see here, can you see we've added C. difficile into this fecal slurry. And whether we add the phages prophylactically, so in advance of adding the C. diff, or whether we add them after, we can see that we completely can cure the infection by 24 hours. So phages, that's really good data because it's showing us that in a mixed complicated infection, the phages are going to come in and just find their right host despite all those other potential bacteria. We also did work where we sequenced the rest of the bacteria within that artificial slurry to show that the phages didn't significantly remodel the microbiome. So the final model that I want to talk about is using big animal models. So this is useful in many ways. So we've been doing quite a lot of work on pigs. One reason is to remove infections from pigs when they're being produced to prevent the buildup of AMR in pigs and then being spread to humans. So a lot of the initial transmission events is thought to happen by the meat that we actually consume, whatever meat it is. So if you can stop infections, this is useful. But I think also by examining how phages work in large animals, there's a lot of useful data that we'll then be able to apply to a human setting. So we did some pig work, and we're just about to do quite a lot more. But this is some data from a small pig study where we had pigs that were given phages in their feed, and essentially so we had pigs basically that just had the bacteria, and pigs that had the phage, and then pigs that were treated. Now, what we showed is that the pigs that were given the phages in their feed in general had lower levels of disease. So this was with a salmonella model. So we can see the red is the challenged and the untreated, and the green is the treated. So this is when the pigs were sacrificed. So it was generally a lot less colonization. And we could also see this manifested in how sick the pigs were. So the animals that weren't treated had really swollen intestines in comparison to those that were treated. And then importantly, if we look at the next plot, we could see, this is now comparing the phage numbers in animals that were just given phages, so that's in blue, compared to those that were given phages and bacteria. And what we can see is that where phages had bacteria, where the pigs were given the phages and the bacteria, those phages were replicating. So one of the major advantages of using phages over a phage product is that we have this self-replicating medicine at the site of infection, and we could see that. So one of the things I think we'll need to do a lot more of before deploying phages is to really understand the pharmacokinetics. So I think we can get lots of information that's very useful from our animal studies to inform that. Okay, so that's a sort of quick romp through all of those models. And as I said, that data is published in a recent paper to show you the different models in a bit more detail if you would like to see. And now I'm going to spend perhaps a few more minutes talking about, this is in a way part 1B of my talk, which is how do we find really good phages from the outset? So we all know when we develop an antimicrobial, it's years of development actually to get something to market, to go through all the regulatory procedures and so on. So if we could sort of find the right phages earlier on, because at the moment what most of us do is we isolate hundreds of phages, then we do lots of tests to work out what the host range is of all of those phages, as I said earlier in my talk, and then we look at the killing, and we can look at all these models. And this is very informative, but it's very, very time consuming. So I've just been thinking a lot over the last couple of years as to how we can speed up this process by identifying sort of phage types, as it were. So if we go back to my botany analogy, if I want to plant a garden here, I'm currently actually at my brother's house in the Highlands of Scotland. If I wanted to plant a garden here, I would select very different plants than if I wanted to start a garden in somewhere nice and sunny, so in a nice part of India, for example. So to plant your garden, you have to understand your soil type and your weather conditions, and then you select appropriate botany that's adapted to those conditions. Okay. With phages, at the moment, we just get loads and loads and loads, and then we do a whole set of screening, but we haven't identified the actual types. And this is what I'd like to do. We're working towards doing that. So here we can see in this picture, it is a picture of it's just a schematic of a Klebsiella cell in the background. But what is real is the blobs. The blob data is real. And what the blobs are all the genomes of all the known Klebsiella phages. Okay, so Klebsiella is an important pathogen. Problems with antimicrobial resistance have been well documented. And all these little clouds are clouds of related Klebsiella phages. Okay, so what I'm trying to do is understand that relationship between the genetic relatedness, so this is based on the overall similarity of their genomes, and the behavior of those phages. So the clouds that I've colored in different properties, the green cloud and the pink cloud, I know that they have different types of genes that are indicative that they may be behaving in different ways. So what I'm trying to do at the moment is relate different behavioral types and different phenotypes with these different genotypes in order to be able to have this concept of a functional type. So I made that sort of analogy with botany, and it's the same with bacteriology. So we know, for example, like some bacteria, we know they're host adapted. If we think about host adapted bacteria, we know that they'll have certain features, or opportunistic pathogens, again, we know that they'll have certain features. So we're not looking for taxonomic relationships, but we're looking for these sort of functional ecological types. And the way that I've been trying to do this is published in a paper I recently published here. So I worked here with plant ecologists and bioinformaticians. And what I tried to do was repurpose a really useful ecological framework in plants. So this is a framework which was proposed in the 1960s by a professor called Grime in Sheffield University. And what he proposed is that all plants could be separated on 3 axes according to how good they were at being a competitor. So that means how good they are at growing and keeping other plants out when conditions are good. So that would be something like a stinging nettle plant. So it grows a lot of big, strong roots, a lot of defenses on the leaves, and it really makes a nice big structure before it starts to flower. And we have other plants that are super good at tolerating stress. So plants that grow on the tops of mountains where there's limited water, for example. And then we have other plants that are very good at coping with disturbance. So plants that are on soil that just gets, people walking through it the whole time, or fire, any type of physical disturbance. There's other plants that are really good at doing that. And what they do is that they're just really good. They go in and they quickly get to reproduction. So the stress plants, what they do, I should have said, they tend to be quite small and very economical with their resources. So this is used widely in botany, and all plants can be classified according to these 3 axes. So some plants are strict one category, and others have properties of both, or indeed all 3. So what I worked to do was see if I could actually use this framework for phages. So it may not ultimately end up being a good framework, but I wanted to test it, and I'm carrying on doing this because actually I think the data I have so far looks really promising. So the way that I've been doing this is, if we think about it, a competitor phage would be a phage that tends to go in and basically it would retune the bacteria to make it have all the things that it needs to be able to make a lot of phages. So it would retweak that host transcription of the bacteria so that the cell can rearrange itself significantly to make a lot of phages. Now, a stress phage in comparison would be a phage that would be able to go into a bacteria and then kind of hang out and hide out if conditions were not good in that bacteria. So it would have a lot of genes to it would go in, and it wouldn't do a huge amount straight away, but as the bacteria was experiencing stress, it would have a lot of mitigating strategies to be able to survive. And then a rudderall phage, again by analogy, would be a phage that would be able to get to lysis quite quickly. So again, what we don't know of phages is what traits to measure. So it's well established in botany which traits to measure on the plants to understand which of these axes they belong to and where we should group them. So what I've used as a proxy, I'm not going to go into this in a huge amount of detail, but just to say what I've used as a detail to start with is the transcriptional profiling of phages. So we know that all phages, they have so much genetic diversity, it's very, very hard to find something in common, but they all go into cells and take over the transcription and get out. So if I just look at the proportion of genes that potentially are operating early, middle, and late, so rudderall phage it just zips in, and it has these late genes. It has the high proportion of these late genes. So I can look at the proportion of genes and then see if phages do separate out under these axes and if this makes biological sense. So from the data we've looked at, only 42 phages, amazingly, I think it's 9,000 phages now and that have been sequenced, but the transcriptomes have only been looked at for 42. So we're trying to expand that in other groups to understand this better. But interestingly, I just wanted to show you that phages do seem to be stratified according to their transcriptional profiles, and what we see does seem to make sense. So phages that live in very stressful environments, shown in green here, they seem to do that. And then other phages, we can see phages on all aspects of the triangle. So I'm trying to develop this further. I'm looking both at the transcriptomics and also looking at the proteomics to ultimately, when we identify genes, as I said, there's so much unknown diversity. I'm working a lot more now with structural biologists to try to get a mechanistic understanding of the proteins that look important. We're also looking at metabolomics to actually get good indications and quick indications of how a phage may be taking over that cellular metabolism. And then this data, we're generating a lot of data, so we need now to use a significant amount of bioinformatics to put all of this together. So that's essentially that part of my talk. In a way, the main thrust of my talk is and the thing I want to leave you with really is that in order to develop phages therapeutically, we really need to have a better understanding of them. By not identifying these functional types, I think it's slowing us up. So that's where I'd like to be. I'd like to be, basically, I mean, we're in the process where together as a research community across the world, we can hopefully contribute to writing this book so we can identify and know which phages will be best to use. Then, having identified those phages, we then need to work out how we're going to put them together as cocktails. So again, this is another cloud showing you different relationships of phages throughout. This is a salmonella cloud, actually. So what we don't know, again, is if we've identified these particular types, we'll hopefully be then able to say, aha, well, we know that if we combine one of these types of phage with one of these types of phage, these will work well together. So I think understanding individual phages is going to be useful. And then that will inform how we make our phage cocktails. So this is my vision of how cocktail development will be in the future. We'll be able to see where all our phages are distributed within our triangle. And these will be, and we'll know how particular phages work in our different model systems, and then we'll be able to then put together these really nice pairings that work synergistically together. So where should we focus now? This is in the way that was the main thrust of my talk, but now I want to sort of talk about how I've been focusing this in specific settings. And I'm talking about now where I've decided to focus in the UK. Because I think in order to get phages closer to being used, we have to use something where there's a clear clinical need that's not being met by antibiotics. So we've been focusing in on urinary tract infections. And the reason for that is that AMR infections are still massively going up in the UK. Bloodstream infections are going up. And the numbers are all here. I don't expect you to read the slides. I just want to make these points that bloodstream infections are going up. But actually, half of all bloodstream infections in the UK are caused by an underlying urinary tract infection. So I don't think in terms of phage therapy development, it's easy to go straight to using phages where you're injecting them intravenously. But if we can prevent the need to do that by treating urinary tract infections, which in themselves are miserable, then we can really, I think, do good things. So we can see the bloodstream infections in the UK. Two of the most important suspects are E. coli and Klebsiella. So we've been focusing in on those two systems. So yeah, I don't need to read this slide out. Essentially, I just wanted to remember to make the point that UTIs are again an increasing problem. Well, I think worldwide really, particularly in the context of the use of catheters. And we do see significant trends in UTIs in terms of the amount of actually AMR strains that are causing them. So what we're aiming to do is to do a clinical trial in UTIs to both treat disease and reduce sepsis. So I'm working, this is my little team on this project. I'm working with Melissa Haynes, who is a medical doctor, as well as having a PhD in microbiology. And she is now leading the sort of patient-facing end of this project. We also have a public health doctor called Marie-Noël Vu, who is really focused on how we map what we do as phage research as to where that need is. So there's no point in us developing brilliant phage products to treat something where there's just no need because antibiotics are considered to be acceptable or are acceptable. They're working efficiently. So we need to identify these cases and these conditions actually where there's significant problems associated with morbidity, mortality, and of course expense. Now, what we've done so far together as a team is we've been selecting the exact patient group that we're going to work on. So actually what we're going to focus in on is recurrent urinary tract infections in females. And that's because we have a population who are very not complicated by other comorbidities. They tend to be a very compliant population. They're just fed up of having these horrible UTIs. And then the things that we're considering right at the beginning are, you know, what the duration of treatment would be and how would we administer them. And how will that then inform the other bits that I've been talking about earlier on in my talk? We'll consider the manufacturing needs and whether we have to do work to consider how phages will be accepted by patients. And then we'll look at, we've been thinking already about what models we will use to collect the relevant safety data. What we've done so far is map out a good cocktail So this is based on identifying the virulence of particular phages. So I could draw your attention to this particular paper published again last year that showed that a very nice way to compare virulence of phages between labs is to integrate the area under a kill curve. So through doing this, we've managed to identify an effective cocktail of phages to treat antimicrobial resistant E. coli and Klebsiella. So having done that, we then put our cocktail together. I just want you to see here from this heat map that the dark blue are where you have effective clearing. So we've got different phages individually and combined. And in our cocktail so far, we can kill fairly effectively many strains, but we still have some gaps. We're currently improving this cocktail by bringing in other phages. And then looking at how the phages combine is also really key. So we will have phages in our mixture that don't kill everything. So this is an example of a phage. If we look in the middle, there's one particular phage here. This is a kind of slightly hard graph to get your head around. Essentially, it's showing the amount. There's a green and a red and an orange dot, and it's showing the amount of phages that, the amount of bacteria that particular phages or phage combinations kill. So one particular phage doesn't do any killing itself of the strain, but what we can see is when we have 3 phages together, it's not limiting that cocktail within this strain. So there's a lot of these synergy experiments that you need to do to make sure your phages work well. So we've been doing that, and we've optimized a couple of robots to help us in this endeavor. And now we're working actually with antibiotics to make sure that they're compatible with current antibiotics that are used to treat UTIs. So we've shown as well that the phages work really well in biofilm, which is where they need to be. So this is also really useful. So going forward, what we're doing with our urinary tract infection work is that we're trying to understand the cocktails and improve them. We are looking at synergies with antibiotics. We've actually now got an artificial bladder, which we'll be using to test their efficacy in a very similar as we can get it setting as they would be working in. Plus, we're also we've just finished our in the UK, the ethics are quite complicated, but we've got ethical permission now to get bits of catheters from people who have infected catheters to show again, to look or to determine which is going to be the best cocktail of phages to use there. So we're working with other people carrying out urinary tract infection trials. So there's a group in Switzerland and in Georgia, in the Eliav Institute, who are doing a trial. So we're working a little bit with them. And importantly, we're working with local doctors who have clinics and are capable of running clinical trials, so we can put our phages into a framework where the people are used to collecting appropriate data, so the phages can be compared to other treatments, which will provide data that hopefully will promote their development. So hopefully I've shown to you that I think it's a really exciting time to be studying phages, but because they don't work in the standard ways in antimicrobial, I think it's really important that we change our thinking with these things into trying to really understand how they work naturally in order to straight away have a really good product before we do the downstream components. I think there's heaps of potential, and it's a really nice time to be studying it because we have got the tools. 10 years ago even, it was really expensive to sequence genomes, but now those costs have come down and down and down, so we can answer more questions. And we have a bioinformatic computing to be able to understand diversity and constrain it. So I just need to very briefly thank my group and the funders that allowed this work. And I wanted to mention at the end of my talk that I am the editor-in-chief for the journal Phage, which is the only journal dedicated to phage research. We publish phage research on all aspects of phage-related work, and we very much welcome submissions from Indian scientists. We had a focus recently on phage therapy in India, and we'd very much like to follow that up with research articles. So feel free, at the moment it's still free to publish in, and it's really quick time from submitting to being able to publish. So with that, I will thank you very much for your attention, and I'll happily take any questions that you may have about any aspect of my talk. Thank you.
Thank you so much, Dr. Martha, for the very informative talk and also enlightening us on the new ecological concept for phages. Thanks to your love for your for botany. We now welcome any questions from our chair and delegates.
Dr. Mata, this is Dr. Vinod.
Hello.
Thank you once again for accepting our invitation and highlighting the importance of getting phages close to the clinical deployment. It was an excellent presentation. I have two questions, and the third one is a submission. The first, so what do you feel like, when do you can predict the phage can be deployed for the clinical use? How close it is?
How close are we to being able to deploy them? Is that the question? Sorry. Yeah, well, I think at the moment we can see there are more clinical trials happening for the later phases, phase 2 and phase 3, and you can see there's a lot more. There's just a lot more clinical interest. I mean, I can see in my own institution, when I started my group in 2007, there was more or less zero interest from my clinical colleagues. And now I have them always bashing at my door, oh, will you help me with this disease? And will you help me with that? So I think the thing that's going to dictate when the time is going to be depending on the amount of effort that's put in. But I think with more and more groupings and collaborations of phage researchers and clinical people with diseases they can't treat, I know of many of these types of collaborations, and I think that you're still talking, I think, probably more than five years, I think, to get something into more routine use. But I think probably there's going to be a much more common compassionate sets of compassionate cases, which will then help build the sort of body of data which will take us to clinical trials. So I still think it's not immediate, because I think there's still quite a lot more work to do. And I think people want to rush, like even my clinical colleagues will say, well, I've got people that are dying, give me phages now. But I don't want to give them phages now. I want to give them phages when we've actually done the work to make sure that those phages are going to work. So
that's the second question is, could you please highlight on the regulation aspect of utilizing phages in UK?
Yes, so the UK is sort of similar really to the rest of Europe in that it's that phages can only be used under compassionate usage when there's no other existing treatments. So there are no routine phage products being used in the UK. I mean, there have been some quite high profile clinical cases. So for example, the case of the young girl who had cystic fibrosis in her replaced lung and a mycobacterial infection. This was done in the UK, and it was done under the hospital, the ethical board of that particular hospital, of Great Ormond Street Hospital. So I'd say that the regulators are becoming increasingly engaged and interested. And one thing I think is really interesting is that the people that regulate our veterinary products they're super interested. And I think we'll probably see products in animals quite soon. So I know of several programs that are pretty advanced. I think you'll probably see phages being used to treat infection in animals, perhaps maybe even as soon as perhaps 3 years, quite soon in comparison. But I think that data, as I say, I think that will help us inform the medical trials, because at the moment, getting regulation right is key. And I'd say in the UK, the regulators are just becoming more aware, and they're giving us quite helpful information about what data they actually need.
Thank you very much. And the submission, I said, even our society to have started the International Journal on Bacteriophage Research in the year 2021. Definitely we look forward for your inputs, in developing and making this journal as an international journal.
Yes,
Martha,
I think
thank
so.
you.
I think there can be really nice synergies with definitely between them.
Definitely in future, I love to hear the case presentation on the phage application from you.
Thank you.
Thank you, Dr. Martha.
Thank you very much.
Sandeep, over to you. Dr. Sandeep, over to you.
Yes, sir. We have a question from the audience. Ma'am, it's from Sashi. She wishes to ask how the phages can enter the eukaryotic cell. Are there reports that phages can enter the eukaryotic cell? If yes, then what is the mechanism?
Yes, that's a very good question. And the dogma has always been that phages cannot be used to treat intracellular infections. But actually, it seems that there is evidence that in some cases, phages can enter cells. So there's been work, for example, with TB and other intracellular models that people have looked at. So you can use something called Rhodococcus equi, which is a horse pathogen that's used as a model for intracellular infection. And the mechanisms are really unclear, but you can certainly, what people have done is encase phages within liposomes, and the liposomes fuse to the outer membrane of the eukaryotic cells, and at least allow the entry into that first through into the cell. So I think it used, as I said, I think it used to be sort of always thought that it wouldn't be possible, and I still think it's probably going to be easier to treat the infection when it's not intracellular. But often in a chronic infection, you'll have some cells that are intracellular and some that are extracellular. And another mechanism that people propose where they think it may work is that if you have a bacteria that's infected by a phage, and then that bacteria is taken up into the eukaryotic cell, it'll bring the phages with it, which will then replicate. So this could be the mechanism. There's various people trying to figure out what these mechanisms are. It's very unclear.
Yes. Ma'am, a question from my side. You were talking about oral phage therapy given to pigs, in which you talked about the decrease in the phage titer in their gut. There are certain challenges which oral phage therapy generally is associated with the loss of titer as they move through the reach down to the intestine. So have you addressed to this aspect? Faced any loss in titer for that matter?
Yeah, no, that's again I've looked at this in different ways in different disease settings. Funnily enough, with the pigs, we just embedded the phages within the feed, and it's making, we can see from our data that they're making their way through the gut, and they're going into the colon. They're managing to survive that passage of the acidic stomach, and they're making it within the feed. Now, when it comes to just delivering something, for example, our C. difficile phages, the same problems have been solved with other medications that need to survive the acidic stomach. So you can just have pH-sensitive polymers that you can either embed the phages in or encapsulate them in, and then you can see that they'll survive that acidic transit and then be released. So we've done this. What we used with C. difficile is we used a commercially available pH sensitive polymer, and it worked really well.
Okay. Thank you, ma'am. Any more questions?
Yeah, I have a question.
Hello. Yeah.
Hello, this is Dr. Rao. I have a question to Dr. Martha. Dr. Martha, it was a wonderful talk. Just I wanted to know about, because we are stressing on antimicrobial resistance and phages as one of the avenues to reduce the AMR. But now, on the other hand, we are talking about synergy between the phage and the antibiotic.
Yes. Well,
so if you use a lower dose of antibiotic along with the phage, don't you think that it will again lead to AMR? What is your opinion on that?
Yeah, no, that's. I'm glad you asked this question. It's a very important question, and I think that often people think about are phages going to, how will phages replace antibiotics? But I don't see them as being a replacement. I think we can use them to extend our useful antibiotics. So there's many different ways that we can do that. So, for example, in some cases we can significantly lower the effective dose of using antibiotics. So then when you're using a much lower dose to treat your infection, you're not causing such complicated spread and build-up of AMR. And I think as well, when you do a clinical trial, it's often not going to be ethical to withdraw standard treatment. So it's very difficult to do a clinical trial where you have the right number of patients with exactly the right profile of exactly resistant bacteria. So if you could have a trial where you're wanting to use phages across the board, and you can just show that, then you don't need to withdraw standard treatment, because the way phages work as I said, they have these lovely ability to penetrate biofilms. And if they're working synergistically with those antibiotics, you would predict that those patients that had the phages would do better than those that didn't. So that's why it's important in that specific setting to be able to show that you don't have that they work well together. I mean, I think there's going to be lots and lots of different ways that we play this. So, for example, there's some lovely work that was done by the lab in Yale by Paul Turner and Ben Chan. And what they showed is that they used phages that deliberately targeted the efflux pumps. So the only way for the bacteria to become resistant to the phages was to lose the ability to pump out the antibiotics. And then they became sensitized to the antibiotics again. So they tend to use phages to treat. And then when the bacteria become resistant to phages, they finish off the treatment with antibiotics. So that's one way of using them. I think depending on the specific setting and the way we want to use them, sometimes we'll use them together, sometimes we'll use like one and then the other. So it's sort of understanding, I guess, the understanding the dynamics and how we can use phages to help us prolong our antibiotics, because we want to keep antibiotics in a way for reserve when we have to use them, but we don't. So I see them as being a very often they will be, I think, a complement rather than a strict alternative.
Thank you. Thank you.
Hello, ma'am. I have one question. I'm Dr. Sangeeta. As you said that we can use cocktail of phages for the treatment of various kinds of infections. But I have read one article, phage ecology, in that it is said that phages in the environment, they can interact with each other. Means
Yes.
they may bring about genetic complementation also. So how this can be possible if you use the cocktail then?
Yeah, phages do in the natural environment. Phages will work sort of. They'll have certain phages have certain relationships with each other, and I see this in the lab. There's certain phages I can't purify because they're reliant on a second phage. So, for example, one of them perhaps helps it penetrate, and then another one provides some of the mechanism for the transcriptional takeover. So I think I think it's again important, a little bit like I was answering the last question. It's important to work out which phages work better together. And I think we don't really have this knowledge so much at the moment, but by understanding the types of phages, we'll be able to say, well, perhaps we need this type of phage to work with this type of phage, and then we see these relationships. And certainly in our work, we've shown that if we use 3 unrelated types of phages with Clostridium difficile phages, we cannot generate complete resistance. So the bacteria can only ever manage to generate resistance to one of those phages, if that, and generally we knock out all of the cells. So I think understanding which types of phages work really well together is really important.
Okay,
Yes,
thank you. Thank you, Dr. Martha, for the wonderful talk. And with this, I close today's event. Thank you, everyone. Thank you for joining, and hope we will join tomorrow once again for a very eventful day. Thank you all.
Thank you.
Thank you, Professor Martha. Thank you.
Thank you all.